Okay, so I think we can get started. Welcome everyone online who is joining today's webinar. We are very excited to talk about FluoroSpot and how it's used in clinical trials today. This is a joint effort between Mabtech and IQVIA laboratories, former immune experts based in Belgium. So we're very excited to host you today, Sophia and Aurélie, and it's my pleasure to introduce you as well. So maybe we can start with Aurélie.
Yes. Hello, everyone. So thank you for joining. My name is Aurélie Mazy. I am working at IQVIA Laboratories in vitro immunology. I have now over 6 years of hands-on experience running cell-based assays, including both ELISpot and FluoroSpot as technologies as a readouts.
Hello, everyone. I'm Sophie Denise, and I support the scientists at IQVIA Laboratories as a statistician.
Thank you both. So during the webinar, please feel free to ask your questions in the chat or in the Q&A session, and we will take them after the presentations. Today's presentations are pre-recorded just for simplicity, so I will soon share my screen and start the presentations. Okay, thank you so much everyone for joining again, and I'm very excited to give an introduction to today's webinar. I will focus on providing a big picture introduction to FluoroSpot assay and how it's used in clinical trials. And then I'm excited to hear from our IQVIA colleagues about how they qualified a FluoroSpot assay for clinical sample testing. So to start with, I would like to introduce you all to Mabtech. We have been around for 40 years now, and we've been providing immunoassay kits and instruments for a very long time now. We have global presence. So the countries in pink that are highlighted in this map is where we have direct sales to our customers. And then the countries in yellow are the countries where we have distributor partners. The company itself is headquartered in Sweden, in Stockholm, and this is where we have the manufacturing facilities, R&D, and the commercial team as well. We also have an office in the US where we have the sales and distributions presence there. But I hope that I'm showing you that we are a global company and that we can actually support any multi-site clinical trials, as well as organizations and companies that have international presence. And quality is really at the center of what we do, and this is really one of our values. And since 2006, we've actually chosen to obtain 2 international quality standards, ISO 9001 and ISO 13485. What that means is that every year we are audited by external auditors, and that also means that our quality management system is very strong. This also helps us to supply to GXP labs as well as IVD manufacturers, and also our instruments and software are compliant with 21 CFR Part 11 as well as EU Annex 11 regulations. We also have robust processes for batch-to-batch testing of our products, but we are a flexible company and we like to provide customizable solutions, meaning if we don't have something in place, if we don't have something on our website, please talk to us and maybe we can develop a custom assay for you or a custom workflow. And Mabtech is really known for ELISpot and FluoroSpot. This is something that we've focused on for many decades. And these 2 assays, ELISpot and FluoroSpot assays, are a gold standard for detecting antigen-specific T-cell responses. Over the last years, however, we have expanded our portfolio and added more different technologies into our portfolio. One of them is a multiplex ELISA platform called ARAPlex and ARA. And we actually did a webinar with IQVIA 2 weeks ago about this technology. So please visit our YouTube channel and check out that webinar too. But today I will be focusing on the FluoroSpot assay and how it's utilized in clinical sample testing. So just to give a brief introduction to the FluoroSpot assay, it's rather straightforward, and that is really the beauty of it. It makes it really suitable for clinical sample testing. So the first step of the assay would be to coat a 96-well plate with capture antibodies that are specific towards cytokines that you're trying to measure. The next step would be to take your patient PBMCs or any other sample type and incubate that with a study-specific stimulus. And if that stimulus is stimulating, the immune cells and the PBMCs that you've added into the plate, then those cells are going to secrete cytokines, and those cytokines are going to be captured as soon as they are secreted. You will then remove the cells away because you don't need them for the analysis later. You can wash them out, and then to detect that footprint of secretion, you can use different detection antibody systems and fluorophores to distinguish the signal and to find out, the footprint that came from this cytokine is going to be green, the footprint that came from this cytokine is going to be yellow, and then this cytokine will be red. So the resulting readout will look something like this, which is very nice readout and pretty to look at. It's a bit like a Christmas tree, but the output of it is really the number of spots per well. And this is indicating how many T cells have responded to your original stimulus. In addition to getting total number of spots per well, you can also look at co-localization of signal and find how many cells have secreted 3, 2, or 1 cytokine at a time. And the cells that secrete more than one cytokine at a time are typically referred to as polyfunctional T cells. This is increasingly more studied, and there are multiple disease models or vaccine models where there have been some correlates of protection that has been found, and so it's definitely very relevant readout to obtain. And a common misconception that we hear from our own customers is that fluorescence detection, or the FluoroSpot, is somehow more complicated than a more traditional method called ELISpot. But as you can see in this chart, both of those assays are very, very similar when it comes to lab work. They are just as simple as each other to perform, so FluoroSpot is not more difficult. The main difference is the detection steps, really, and one thing that FluoroSpot avoids is this time-sensitive step in ELISpot, where you would have to incubate the substrate for a very specific amount of time, because if you leave it for shorter or longer, that can really affect your readout. So the FluoroSpot doesn't have that time-sensitive step, so it actually makes the assay also a little bit less stressful. But of course, the beauty of FluoroSpot is that it provides a lot more data from a single well, so you can multiplex from 1 to 4 analyzed per well. But both of these assays have been used heavily in clinical sample testing, but we have seen from our customers that there is an increasing interest in using FluoroSpot in clinical sample testing because you can get so much more data from a single well. And I want to show you a few examples of how FluoroSpot can be beneficial in clinical sample testing. The first example I want to show you is to do with a vaccine clinical trial. This is a phase 2 clinical trial where they wanted to look at T-cell responses towards the SARS-CoV-2 vaccine. In this case, they wanted to look at both Th1 and Th2 responses, and this reflects a wanted and unwanted immunogenicity. So in your vaccine responses, you don't really want to have IL-4 or IL-5 responses because that could indicate that there could be some allergic responses to your vaccine, while the Th1 responses that are driven usually by interferon gamma is something desirable, and this is something that you want to induce. So in this study, they were able to multiplex interferon gamma and IL-5 together and found that their vaccine induces a robust Th1 response, but there was no Th2 response induced, which is, good news for a vaccine trial. Another example I want to give is to do with an autoimmune disease, type 1 diabetes, and Th17 cytokine is something that can be included in autoimmune disease context. So in this case, this is a trial with a monoclonal therapy where they have multiplex detection of interferon gamma, IL-17, and IL-17F in the same assay. And they were following patients before and after the treatment. And what they found was that there was no effect in interferon gamma responses throughout the treatment, but they have found that there was a decreased pro-inflammatory IL-17 responses in those patients after treatment. So if they have only included interferon gamma, they would have missed that therapeutic effect or that effect on the T cell populations that their monoclonal therapy had. And the last example I want to show you is regarding cytotoxicity measurement. So this is, of course, very relevant in immuno-oncology, but also viral infections, for example, HIV. And the paper that I'm showing here, this was a personalized mRNA vaccine. where you will basically measuring using interferon gamma ELISpot assay to find first which antigens these patients are responding to in the neoantigen vaccine. But once the responders have been identified, these researchers wanted to look if those T-cell responses induced by the neoantigen are polyfunctional. So they used a triple color FluoroSpot assay, interferon gamma, IL-2, and granzyme B, to multiplex those cytokines and to confirm that actually indeed those responses were polyfunctional. So I hope I convinced you with a few examples that FluoroSpot can be beneficial in clinical sample testing. But now I want to shift gear a little bit and go in a bit more detail on, what does it take and what is required to set up a FluoroSpot assay for clinical sample testing. So today I will focus primarily on the critical reagents and instruments, and IQVIA is going to talk about the samples that you need to obtain, and they need to be high quality. How do you make sure that your operators are qualified, and also what kind of experiments you should be performing before you use the assay in a clinical trial setting. So I want to start with the critical reagents aspect. And as I mentioned in the beginning, Mabtech creates pre-made FluoroSpot kits that come with everything that you might need to run the assay. So those kits include the plates, they include the antibody pairs that we have optimized and know that they will give you good quality, sharp spots. Those kits also include conjugates, substrate, enhancers, any other buffer that you might need, as well as the positive controls. And of course, there are different formats that are available of our kits. And some of the kits come with uncoated plates and some of them come with pre-coated plates. And a lot of our customers that use our kits in clinical sample testing, they actually always go for pre-coated plates. And this is because you can save time in the lab, so you don't need to manually coat the wells yourself with the capture antibodies, but you also reduce the assay variability that can be coming from that step. So at Mabtech, we actually have automated process for pre-coating the plates. So we ensure that, it's a very robust process and it's done with liquid handlers. So it's not done manually. And we also have a proprietary technology that stabilizes those capture antibodies on the plate, meaning that the shelf life of these plates is high, very extended. We also have custom plate coating services. So if we don't have a cytokine combination on our website that you would like to have, we can actually create a custom kit for you as well. And actually, the quality of our pre-coated plates has been highlighted by the Vaccine Research Center of NIH. They recently published this paper that I highly recommend you check out. Here, they've automated the entire FluoroSpot assay from the beginning to the end, and also actually integrated our IRIS-2 instrument into the Thermo Fisher Orbiter to make the data acquisition faster. But if you look at their results when they tested FluoroSpot, and their assay precision data is extremely good, and that also showcases how robust our pre-coated plates are. Another thing that you might not be aware is that we actually have peptide pools. So peptide pools is definitely one of the critical reagents. You want a positive peptide pool control that will stimulate most of your donors. And it's a very biologically relevant control, especially if your test article is peptide pools as well. So we recently launched a new version of peptide pools that can be used as positive controls, and they are called Cephras. And those peptide pools have broader pathogen coverage than the typical ones on the market. And they also have a more diverse HLA coverage. So you will definitely be picking up more positive responses from your donors with our positive peptide pool control. So there's more information on the website that you can find. And the last part I want to cover today is our instrumentation. This is obviously as critical as the assay itself. You want to acquire data in a robust and reproducible manner, and we have created two instruments, Mabtech Aster, which is an ELISpot only instrument, and Mabtech IRIS-2, which is a ELISpot and FluoroSpot instrument in one. And today I'll focus more a little bit on the iris, but generally what we wanted to do with our instruments is to make them as easy as possible to use. And also we wanted to reduce the variability that can be introduced by the operators having to adjust hardware settings every time that they need to run the plate. So for this reason, what we've chosen to do is we've chosen to have a fixed focus and preset exposure times for all of our assay principles. And the instrument also has a self-calibrating XY table. And because we have taken this away from the operators, what we've seen is that the operators with different experience levels can obtain very similar starting point when it comes to spot forming units. And in comparison to other systems on the market, where you have to fiddle around with a lot of hardware settings, and that really can affect, the starting point of the spot numbers that you have depending on your experience level. So this is something that we think we really improved in the field. And that also means that if you have multi-site studies and want to make sure that your instrumentation and data that you start with is as similar as possible, iris is a great choice for that. And our instruments are also designed to comply with CFR 21 Part 11 and Annex 11, as I mentioned before. Another thing that I want to mention, and this might be relevant definitely relevant for our customers that work in a regulated environment, is harmonization and automation services. So our instruments, because we produce them obviously ourselves in-house, and because of this nature of fixed focus and preset exposure times, we can actually harmonize instruments to be very, very similar to each other. So for example, if you want to have your primary and backup instruments to be performing as closely as possible, we can provide that service and harmonize the instruments with each other. We also have automation solutions, as I mentioned. So if you have many, many plates that you run per week and don't want to sit at the instrument and manually feed the plates in, we have a automation solution with a Thermo Fisher's orbiter that we can set up for you. And one more thing that I wanted to mention, I already mentioned before, but it's regulatory compliance. So we have spent a lot of time and effort to make our software as compliant as possible, because we know that in order to use these instruments in a regulated environment, we need to have good data integrity, traceability, and documentation. And you can read a little bit more about how we comply with these regulations on our quality website. So I will leave a link here. And in addition to the compliance, we've also developed a performance qualification plate, which can allow you to track the instrument performance more frequently than the annual service that we provide. So for example, you can choose to monitor your instrument every 2 weeks or every month to just have that peace of mind that instrument is performing, and you can produce documentation that it is performing well. And we really work very closely with our customers, and we take your feedback in to enhance regulatory compliance and to make sure that the data analysis workflow is as easy as possible and is as, that you have a good experience with it. So with that, I just want to say that we are here to support you. We have a lot of scientists in our company that can help you. Most of them have background in immunology or other relevant fields. We also host advanced ELISpot and FluoroSpot courses that you can sign up for. We have a lot of webinars on our website, as well as we have a knowledge hub with a lot of tech notes and protocols that you can go to for inspiration, as well as a publication database that outlines all the publications where our products have been used. So thank you so much for listening today, and I would like to leave the floor to IQVIA colleagues now. And thank you.
Thanks everyone for joining today. What we want to share with you is actually how we qualified our FluoroSpot assay at IQVIA Laboratories in vitro immunology with the support of Mabtech. To begin with, just a few words about us. We are located in the French-speaking part of Belgium with dedicated laboratories, offices, and cryostorage. As you can see, we have multiple technologies available, but including immunoassays using ELISpot and FluoroSpot as a readout. Our focus is on in vitro translational immunology to address both the safety and the efficacy of drug candidates. Our capabilities are organized into several service lines, which can potentially all involve FluoroSpot assays as a readout. However, in our experience, in most cases, this technology is often used in ex vivo settings for exploratory or secondary readout in clinical trials. Because as already mentioned by Renata, yes, the FluoroSpot can be used in clinical trials for immunomonitoring and actually is widely used to evaluate vaccine immunogenicity and immunotherapy responses. It will allow you to measure antigen-specific responses at the single cell level and will give you both a qualitative and quantitative immune profiling, as well as supporting your efficacy assessment. Again, unlike ELISpot, the FluoroSpot can measure multiple cytokines simultaneously, which will really allow you a deeper characterization of your T cell phenotype and functionality. It is usually incorporated after proper assay qualification, which will demonstrate its high sensitivity for low-frequency immune cell detection and also its strong reproducibility. And this is also indeed something that we have experienced to be a suitable assay for multi-trial samples because of its possibility for standardization across labs. Now, when it comes to designing your FluoroSpot assays, there are several key factors to take into consideration. And the first one being the PBMC sample quality, so the quality of your input material. And of course, the first thing that comes to mind is the cell viability. And achieving high cell viability requires to have an optimized workflow and procedures for PBMC isolation, freezing, shipping, but also downstream handling to ensure that the samples will remain suitable during the whole process. But next to viability, the cell functionality is actually equally important because the functionality integrity of your cells will ensure that these reflect the true biological activity and enables you to have reliable and meaningful measurements. And just to give you an idea, in our labs, we want to recover, of course, high functional PBMCs, and for that, we typically perform the isolation within 6 hours after the blood withdrawal. Now, once you've collected these high quality samples, you will be able to achieve a higher reproducibility, which will allow you to standardize and harmonize your assays across runs, across laboratories, which is again an essential requirement for multi-center studies. And finally, with having this proper sample quality, you will also have stable measurements when it comes to comparing a baseline sample to a post-treatment sample, or to even have repeated measurement of the same sample, for example, the baseline, and this throughout your entire study duration. Then, in addition to ensuring a high sample quality, it is also essential to include the relevant and appropriate controls, because these will be very critical for demonstrating that your assay is actually functioning properly and that the resulting data are valid and can be included in your final data sets. The controls will first confirm that your PBMCs are functional and to really help you avoid including false negative findings in your final data set. They will also help you to distinguish the biological signal from the background noise, which is of course very important when interpreting low frequency responses. They will also enable you to early on detect any technical failure that could have happened during any step of the assay, and then will prevent you to enter this compromised data into the final study data sets. They will also help you ensuring the reproducibility of your assay, whether it is across sites, across operators, time point, and so on. And with these proper controls, you will allow to have normalization and also fair comparison between baseline and post-treatment samples across different treatment arms or across different time points over your study duration. And for the qualification that will be presented later today, we have included 3 different types of controls. There was first the reference control, which is basically the untreated cells. Then we have a negative control being the buffer, and then several positive controls. We had 2 different peptide pools provided by Mabtech, the SNMO and the CEFRAS that Renata was mentioning, and which would basically cover for COVID-19, EBV, influenza, CMV, and others. And we also included the anti-CD3/CD28 as more as a self-functionality control. Another key element also already mentioned by Renata in the design of your FluoroSpot is really the selection of your analytes, because it is indeed a choice which is highly study specific and that should really always be guided by the scientific question you want to answer. And by combining these multiple cytokines, you can really already tease apart different types of immune responses, whether it is more a CD4 or a CD8 response, whether you are more in presence of a Th1 or a Th2 profile, early versus late responses, and so on. And your analyte panel can also really depend on your therapeutic modality, and it may differ whether you are working with vaccines, immunotherapies, or if you aim to assess autoimmunity, inflammation, or cytotoxicity. So here for each topic, let's say we have included some cytokines that that can be of interest to address your scientific question, and you can see that they can be very different depending on what you want to know, basically. And finally, another important aspect we wanted to talk to you about today about the assay design is the definition of your acceptance criteria. And this should be done early on, really during the initial planning phase, and then should be implemented for the routine use of your assay for your specific study. And they will really allow you to validate the data generated in each individual run and to determine whether or not these results should be included in your final data set. These criteria can include the PBMC post-O viability recovery, but also the performance of your cell functionality control as well as negative control. And all these criteria will help you to maintain reproducibility across your different runs and labs. And to support this, you might also consider to measure the baseline sample in every run, but the baseline sample material can be limited, of course, and so another option is to include a healthy donor in each run as a reference control across runs. And the key question is, of course, is how to set these acceptance criteria. And what we do in practice is to typically derive these from the data generated during our assay qualification. And so this is the work that we have performed at IQVIA, and I will now hand it over to my colleague who will actually present our FluoroSpot qualification results.
Thank you, Aurélie. Before going into the qualification data, I would like to spend a few slides on some general concepts, starting with the difference between qualification and validation. Although qualification and validation rely on the same type of experiments and also evaluate the same parameters, they differ in depth and rigor. Qualification mainly attempts to describe assay performance, while validation is really a formal evaluation of assay performance. As a result, validation is typically more extensive, and a practical difference between the two is that for validation, acceptance criteria must be predefined, and the performance will be evaluated against these acceptance criteria with a formal pass-fail outcome. For qualification, this is more flexible. Qualification still aims to answer the question if the assay is fit for purpose, but this evaluation is less stringent, more descriptive, and no predefined acceptance criteria are required for qualification. The next logical question is, of course, when to do what, qualification or validation. And for this, the concept of fit for purpose is key. The extent of the assay evaluation should be driven by the intended use of the data, the stage of development, and the impact it has on decision-making. So typically in early phase clinical trials, so phase 1 or phase 2 trials, for secondary and exploratory endpoints, qualification is sufficient, while when moving to phase 3 trials, where primary endpoints are evaluated, a validated assay is necessary. Moving to the parameters that are typically evaluated for a FluoroSpot assay, there are many parameters like precision or linearity that are very standard across different assays, also for a FluoroSpot assay, but there are two that are a little bit different in the context of a FluoroSpot assay. The first one is accuracy. Accuracy is a key parameter for many assays, but it's not applicable for a FluoroSpot assay simply because we have no true reference values for cytokine-producing cells. Another very important parameter for a FluoroSpot assay is the biological responsiveness, measured with the suitability controls. As it's a functional assay, the sensitivity is not only defined by the analytical sensitivity. So low background gives the analytical window to detect responses, but if the cells are not functional, the overall sensitivity can still be very poor. So that's why it's very important in these types of assays to demonstrate your sensitivity based on these suitability controls. Before going into the actual data, I want to just discuss some general concepts that are important for the design of a qualification experiment. So there are many ways to define the design of qualification experiments, but 3 principles are very important. The first one is that some of the parameters are truly dependent on donor. So, for instance, background and responsiveness are typically donor dependent, and for these it's very important that sufficient number of donors are included. For other technical parameters, these can be evaluated on less number of donors. So for instance, in our qualification, we evaluated background and responsiveness on 10 donors, but then precision and linearity only on a subset of 3 donors. The second important concept is that the control should cover the entire assay range. It's not sufficient to document the performance only on strong positive controls, as this is not representative of what can be expected during routine testing. The evaluation should also cover representative experimental conditions that can be expected during routine testing. So the evaluation should include multiple runs, operators, and any other factors that may vary during routine testing. It's important to note that it is important for evaluations other than precision alone. So for instance, also the background or your sensitivity should be evaluated across multiple days. It does not mean that it has to be repeated across multiple days, but the evaluation should at least be spread across multiple days. The next slides show a brief overview of each of the different parameters, and we start with the LOB/LOD data. So what you see here is the SFU counts. For 10 donors, it's all the unstimulated control that you see here for the 3 cytokines that were evaluated in 3 different independent sub-experiments. You see the results here for the LOB and the LOD. I added the technical definition of LOB and LOD here. What is important to realize is that for a FluoroSpot, these estimates like the LOB and LOD are not used as thresholds for positivity. So typically, positivity is analyzed via, for instance, a fold change or a delta SFU, always normalizing versus a donor-specific unstimulated condition. We will come back later to that when discussing the biological responsiveness. If you look at the actual numbers, we see that there is a very big difference between the cytokines in background levels, which is a representation of the different biologies. So the different cytokines are secreted by different cells. They're also regulated by different pathways, and this shows in different background levels. This slide shows the overview of the precision. So we evaluated precision by repeating a subset of 5 donors with the different simulation conditions across the 3 days. In our report, we have a detailed evaluation in function of signal. Here we present the overall estimates. You do see that there is a selection to exclude the very low signals. And that is just because inherently CV estimates of very low signals close to 0 are very unstable. And that is why you see the selection here for overall CV only for certain positive signals. Also here, we see very clearly that the estimates are cytokine dependent. So you see here the precision based on the actual SFU estimates. Here you see the precision based on the delta SFU. So after normalizing for donor-specific background in each experiment, and we see that in both cases, the precision for TNF-alpha is much different from the other 2 cytokines. Also here, this represents a different biology, where some cytokines, like for instance, the TNF-alpha, can be more sensitive to these small uncontrolled variations during cell cultures than other cytokines. On this slide, we see the evaluation of linearity. So for linearity, we evaluated two positive controls, so very strong positive control, the anti-CD3, CD28, and then a more moderate positive control, the Cephras, in different donors at different cell dilutions. With the CD3, CD28, we are able to reach the upper limit of linearity, while then with the Cephras, we are able to reach the lower limit of linearity. Important to note is that these limits of linearity are context specific because the instrument setting can impact the limits of linearity. Therefore, it's important to evaluate this based on relevant controls, and then based on these results, the cell densities can be selected for the clinical testing, which provide results in the linear range for your relevant control. This slide shows the results for the biological responsiveness, so the suitability controls to confirm that the assay is sensitive. So you see here again the CFU counts for different conditions. We have the blank reference condition, 2 buffer condition, then a strong CD3, CD28 positive control, and then some more, I would say, biological relevant controls, the Cephras pool at 2 concentrations and the SNMO pool. We evaluate this for 10 donors across 3 independent experiments, and we evaluated sensitivity as a number of donors that result in a positive response consistently across the independent evaluation. So we consider the response detectable if a DFR test gives a significant result. So the DFR test is a kind of statistical test that evaluates if the counts in the test condition are significantly higher than the counts in the reference condition. For those who are not familiar with the test, I put the reference here. So there is an online tool available to perform this test, but at IQVIA laboratories, we have an in-house implementation of this test, which is also standardly included for routine testing. So here in this table, you will see the results. So these are the number of donors that consistently show a positive response based on the DFR test for each cytokine and for each condition. So you see that the sensitivity to detect responses is very high across these different conditions and cytokines. So even for TNF-alpha, for which we have seen that there is a high background and also relatively high variability, we are still able to detect in all donors, or almost all donors, consistently across experiments, these significant responses to the different stimulations. Based on this, we can also conclude that, for instance, the Cephras pool is a very appropriate control to use in routine testing, which can be really applied to individual donor levels, because we really expect that each donor will show a response to this condition. As robustness, we evaluated the effect of different reading times on the SFU counts. Also, this evaluation is context specific. So in this specific situation, the plates were kept at room temperature in the dark, and the settings were optimized for a readout after 24 hours, and they were not changed in the course of the time period. How we evaluated this is that we calculated per well the ratio at each reading time versus the corresponding result of the same well at 24 hours. And then we calculated overall wells a geometric mean of this ratio with the 95% confidence intervals, and that's what you see here. So the line represents the geometric mean ratio, and then the shading, the 95% confidence intervals. So we do see that up to 1 week, so the second time point in this graph, we see no or only very minimal effect on the spot counts, demonstrating that under these conditions, we can read the plates within 1 week without changing the settings. It is also important to note that also this evaluation is highly context specific, so not only, of course, specific to the storage conditions, but also, for instance, it can depend on the lead filters that are used, the cytokines and the assay conditions, as also the spot morphology may impact how they are impacted over time. As a last slide, I just summarized the performance of polyfunctional T cells. So We define this as cells expressing at least 2 cytokines. So they show excellent performance. So you see very low background in the reference conditions and the buffer condition. We see good precision with intra-assay CVs below 30%. And we can detect all suitability controls consistently across the donors. These are especially interesting, as Renata already introduced at the beginning of this webinar, as they may be correlated better with the clinical response than individual positive cells. So that was an overview of our qualification experiments at IQVIA Laboratories for the FluoroSpot assay. And now I will give the word back to Aurélie for the key takeaway.
Thank you, Sophie. So indeed, just some key takeaways to conclude this webinar. The FluoroSpot assays are indeed widely used in clinical trials for immunomonitoring and measurement of antigen-specific immune responses. The FluoroSpot technology also allows for multiplexing of cytokine, which is a better way to address your scientific question. And when it comes to designing your FluoroSpot assay, there are important parameters to take into account. First, PBMC sample quality, which includes both the viability and functionality, the inclusion of relevant controls to confirm that the assay is fit for purpose, the selection of your analyze depending on your scientific question, and the definition of your acceptance criteria guided by your qualification assay. And to design your qualification assay, your qualification experiment should really be representative for your specific study. And you should take into account that each parameters can really be cytokine dependent with underlying biology. And if you would be multiplexing cytokines, another important thing to take into account would be the polyfunctional T cells, which in our qualification showed low background, good precision, and high sensitivity. So with that, I would like to thank everyone for joining this webinar today.
Okay, thank you so much for your presentations, Aurélie and Sophie. For everyone who's listening, please raise your questions in our Q&A section or in the chat. So we can only take the written questions, so please write your questions in the chat. I know that some of you have a hand raised, but because this is a webinar, we will only answer questions from if they're written in the chat. But to start off, let's talk about, I think, one of the maybe urgent questions, or someone while we were listening might have a question about the TNF alpha. And a lot of our customers have also seen the same issue with TNF alpha, that you have quite high background levels. And you already mentioned, Sophie and Aurélie, that background levels are always cytokine dependent. We have seen that monocytes can secrete TNF alpha, and of course they are part of the PBMC compartment. But what are your general reflections on TNF when it comes to FluoroSpot, but also other methods?
I can maybe, yes, just start with a statistical perspective of indeed a result that has relatively high CV. So that is the background is of course still relative for FluoroSpot. It's a high background compared to the other cytokines, but we still see a wide window to see increases with a positive stimulation. But we also see, of course, the high CV. So with the high precision, we can typically still balance that with high number of replicates. So a high CV does not mean that the data is unreliable, it's just imprecise. So increasing the number of replicates can still give you stable results. So that's one potential way forward. The other is, of course, to move away from maybe a full quantitative outcome and move more towards a qualitative outcome. As you saw during the presentation, we are still able to detect consistently across experiments in each donor positive responses to different stimuli. So even with the high CV and with the high background, we can still make the distinction between a response or no response. So that would be, for instance, another way to deal with the high CV that we see with TNF alpha.
Good, thank you, Sophie. Do you have something to add, Aurélie?
Yeah, just maybe to to add, sorry, that indeed we can probably see a higher background with TNF-alpha, but that can still be widely used, especially if it's in combination with other cytokines, such as interferon gamma, just to look at polyfunctional T cells, as you were mentioning, just to be a bit more guided towards whether it is a polyfunctional response or just single secreting cells. And even if it seems a high background compared to other cytokines, I still believe this is rather highly sensitive if you would compare it to other cytokine readouts, such as ICS, for example. We are still talking about a few hundred cells in 1,000 cells which were seeded. So that's still pretty low still if you would compare it to another technology.
Yeah, great. Thanks both. So we have quite a lot of questions coming in, so I think we can jump right into them. One of the first questions we had is about the instrument harmonization and what parameters or aspects of the instrument are harmonized. So this is a great question. I didn't go into detail on this question during my presentation, but what we're doing when we're harmonizing instruments is typically setting the same focus and the same exposure times for all different assay principles, which means that we can get the readers to perform as similarly as possible. And typically what we can achieve in terms of spot numbers is less than 5% CV variability between the two instruments, or more than two instruments. So hope that answered your question. Let's see, we have some more good questions. One of them is to you both, Aurélie and Sophie. How do you determine the number of samples to include for each experiment? I think this is a great practical question that you may answer.
Well, I guess maybe I can answer on the practical side of things, because it can vary on what is the manpower that you have available, what are the samples that you have available. For example, if you know that your baseline sample would be limited and that you have an option to test all the time points together with that baseline sample, I would highly recommend to combine all these samples together in one run. If you have that baseline sample limited, if this is not feasible because the experiment becomes too big or your operators do not have sufficient experience to run such a big experiment, I would then recommend multiple runs. But as we mentioned, always then to at least include that baseline sample or reference sample along in every run. I think the most important is that you always need to be able to make a fair comparison between the runs, just to make sure that if you see an effect from one time point to another, this is a true effect and not because the variability that you can have between two runs.
Great, thanks, Aurélie. Is there something you would like to add, Sophie, to this question?
Yeah, I think that indeed Aurélie covered that the ideal design depends on practical limitations, and then also, of course, the design of the trial, between which samples there should be a direct comparison if it's more longitudinal or between groups. The number of replicates per condition is, of course, another question. I don't know if that's also maybe meant. So that really, of course, it depends on the precision of your assay, how many replicates you need, and also on how you will analyze the data. Because, for instance, the DFR test that is relatively, I would say, commonly used in FluoroSpot data, there you do need at least 4 replicates to be able to to perform the test. So if you are less than 4 replicates, it's not even technically possible to get a significant result. So that's also important, of course, to consider upfront how you want to analyze the data, with what test. There are alternatives if 4 replicates are not possible. There are definitely also good alternatives to the DFR test. But if you want to use the DFR test, you should take that into account in the number of replicates that you test.
Great, thank you both. There's a very similar question, but might have a slightly different angle to your responses. So how can I validate an assay when I have a limited number of samples? What is the minimum number of samples required? You touched on that a little bit. And is it the cytokine dependent? And do I need to validate each kit individually? There's multiple questions in this one, but maybe we can discuss at least parts of it. For example, one of them is definitely whether you should validate each kit individually. And from our perspective, yes, they do have different performance levels. So Sophie and Aurélie, if you were to now develop a different FluoroSpot assay, let's say interferon gamma, IL-2, and TNF-alpha, where only one of the cytokines is exchanged, how would you approach this?
I think we can really use a rather limited setup. In the past, what we've been using in such case with indeed limited samples is rather to include a full qualifications, is simply to perform what we called dry runs, using so very limited sample, and also to compare that performance of that sample with a healthy donor, so for which we know how it should perform, and include control conditions, but also some of your test conditions, just to know, is there any background with that specific FluoroSpot kit or cytokine combination? What is the response of one clinical sample compared to a healthy donor sample towards the control molecules? This kind of setup, and that's much more limited compared to a full qualification. But at least it gives you an idea already on what is the outcome that you can expect with the specific cytokine combination, with the specific PBMC isolation procedure, with your specific conditions.
All right, thank you. Thank you, Aurélie. So we have one actually really interesting and practical question as well. If you measure a patient in multiple assays with one shared sample, so let's say always including the baseline, baseline sample, how do you standardize across assays to keep things quantitative? For example, what do you do if the results for the baseline control are not consistent between assays?
So there are different ways to deal with that. So I would say that especially in this assay where most of the time the data is analyzed and not as the more simple assays where you just say threshold positive or negative, but you really do a statistical analysis where you take into account all the replicates of the stimulated condition versus the unstimulated condition. A logical way to deal with this is to do a complete analysis of all the data where you also include the run in your analysis, for instance, as a random effect. So that's really then in the statistical analysis, clinically actually, that you take into account that data. So that would be, I would say, the most complete way of evaluating that. So not necessarily treating every run separately and have there a quantitative assessment, but really take into account all the data and then all the experimental factors. And then automatically, of course, if you include the run as a random effect, you have the normalization for run, but also, and that's very important, you take into account you take along the uncertainty. If you would do a manual normalization, then you don't have the margin of uncertainty around it anymore. So you pretend like you have normalized perfectly, which is not the case. If you take it really into account in the analysis as an experimental factor, your final readout or outcome, main outcome, also considered the uncertainty related to this estimation of a run effect, which is also, of course, only measured with a margin of uncertainty. So definitely, as Aurélie already said, it's ideal if you can keep your main comparisons within the run so that you do not necessarily have to deal with that. But of course, that's not always possible, especially not if you have longitudinal follow-up. It's not always possible to have, for instance, all the results of the same patients in one assay run. But if that is possible, I would say that is always definitely the preferred way. But otherwise, I would say that the best way to deal with that is really to take it into account. You start with all the raw data and all the experimental factors in your final analysis.
Thank you, Sophia. That's a really good answer. It is complex because it is cell-based assays and you do get variability even in your baseline control. So this was a great question. I think very relevant and practical. So thank you. Thank you for this question. So we have a few more. I'm just trying to see which one came in first. So one of them is about actually more preclinical application. One of the scientists here is working with animal studies, and they want to perform FluoroSpot using spleen samples. So maybe we could discuss how is that different from human PBMCs, how much, also maybe we could discuss a bit how much qualification needs to be done. Is there any differences between clinical sample testing, for example, and is it more expensive with FluoroSpot, and how expensive is it? But this is maybe more of a specific question. So we can start with the differences between the splenocytes and PBMCs and the important considerations that from your experiences you've seen are important.
Well, yeah, to first answer the question, it is indeed possible to have FluoroSpot using the spleens of your animal. And we've done that in the past. And I would say they're a critical step compared to, well, it's a bit the same on the PBMC isolation. You need to isolate these from the blood. And for the spleen, the critical step would then be how you dissociate your spleen and how do you recover your splenocytes, basically, which can potentially have an influence on the background you would have if there's a lot of stress on the cells during that dissociation and collection procedure. But this is definitely feasible. And in the past, in our hands, we only use, if I can call it like that, mechanical dissociation to recover the cells, proper filtrations to be able to have a clean cell preparation. And then we were able to measure quite some specific responses using the FluoroSpot kits from Mabtech as well. So I wouldn't say that the performance is as good as with human PBMC samples, but you can still maintain this high sensitivity for that kind of readouts. Regarding the cost, this is indeed not something I can really comment right now, but I would still believe that this is an assay which can widely be used in this application, because as Renata already mentioned, this is an assay which you simply have to plate yourselves in the FluoroSpot plates, and then this is quite of a straightforward readout, which can be really easily harmonized compared to other readouts. So yeah, I would still think that this would be the best way forward for your clinical trials.
Yeah, you can really get much more data from FluoroSpot from a single well, which is the major advantage, and you can save on sample and the price, in terms of consumables, it's only incremental increase when you start multiplexing. So if you do single plex FluoroSpot versus ELISpot, the price is very similar of consumables, and then you have a slight increase in price as you add on more cytokines. But contact our sales teams from IQVIA and Mabtech and for that specific question. Okay, so I also have a question about IRIS2 parameters. And one of the questions is, IRIS2 parameters matter most in the readout of negative controls. How were these parameters established and at what level? So I can just start a little bit that with the IRIS2, there are two parameters that you need to change, only two of them, because as I mentioned, we have fixed focused and fixed exposure times. So the end user only needs to change the size and intensity sliders. And of course, setting those parameters can be a bit challenging because there isn't any way to kind of set the settings in a maybe, let's say, a statistical way. There are a few approaches, but you usually look at the positive controls, at your test article, and your negative controls, and you find settings that don't pick up too much background from the assay noise, but still pick up your strong responses. But I want Aurélie and Sophie to add a little bit on how you yourself established the settings and what approach did you take, and yeah, any comments you have on this.
Yeah, well, for the so the qualification experiments that we've done here, we simply use the predefined settings. But important to say is that if we would change the settings for one donor, one plate, let's say, we will try to keep it consistent across the plates of that experiment, at least for the samples from the same donor. But usually we are quite satisfied already with the predefined settings. An instance where we change it is that if for some reason there was a bit less performance of the fluorescence enhancer, so the last step of the development, and that you can still see that you have a rather specific spots that are not counted, there we would try to see, do we get better results if we would so decrease the intensity or the size, the size inclusion criteria there, just to see if you can have the same output or not. But usually, even in such instances, we would go back to the predefined settings.
Great, thank you, Aurélie. Yeah, so our instruments provide a good starting point, but of course you have the flexibility to change depending on your spot morphology and how your assay actually looks. And every assay is slightly different, and you might have slightly different background levels, slightly different spot size and spot intensity. And there was one question in the chat about whether you can look at the spot size and intensity, and that's definitely something you can do in the software. So I think we also have another readout called relative spot volume that measures relative secretion at a single cell basis. So this would be interesting if for example, if your test article can induce differential effects of secretion at a single cell level. That would be an an interesting readout to also look at, in addition to the just numbers of spots or frequencies. But I think I could start a discussion and ask a few questions about your processes, Sophie and Aurélie at IQVIA. So let's say a client would like to place a study review for a FluoroSpot assay. I would like to hear from you. Is there any additional testing that you would be performing for the study-specific samples, or any other additional experiments that you would be conducting in addition to all the experiments that you have already done?
Yes, indeed, we can touch on that. So the entire qualification that we presented today was done with our in-house isolated PBMCs from our biobank. But when it would come to samples from a clinical trial, we would highly recommend to still perform some experiments prior to running the entire readout on these precious samples. And these additional experiments that we would be running, we call it dry runs. And this is basically to assess what would be the performance of your PBMC samples from your clinical trial compared to the results we have obtained with our in-house PBMC samples. When it comes to the performance of the positive controls, but also some of the test conditions that you would include in your runs later on, just to see, do we detect any background responses that we need to tackle first? Do we need to adapt the cell numbers that we put per wells, or these kind of parameters? So that's typically something that we do prior to running an entire trial, I would say, just to be able not to waste any precious samples in these preparation steps.
Yes, absolutely. It's very critical work before you jump right into many samples at once. So thank you, Aurélie. We do have a question coming in. So as part of a qualification validation, do you assess whether the responses from your multiplex FluoroSpot assay in clinical samples are comparable to the responses assessed in the ELISpot assay, or FluoroSpot assay where only one analyte is evaluated?
I believe this is something that we have never tested. I don't know if you remember, Sophie, but usually if we would qualify a kit, we would do it for each kit. But I don't know, Sophie, if you see any example where we did it differently. I can't recall any, but...
I think indeed, if we would design our own kit, then in development of a kit, that would be extremely important. I think it is definitely added value in a full validation. It, of course, increases the workload a lot if you have to then also test your samples on a different assay. So I would say that is really a risk assessment based on how well the kit is developed already and how much you still develop in-house. And then also, of course, with the stage of the development, if it's very critical, it's definitely useful information. But in our context, indeed, with the kits that we have used and with the context that we have used, then we have not done that.
Yeah, thank you both. And I can add to this question as well. I think it's quite common from our customers, if they are switching from ELISpot to FluoroSpot, to be in evaluating the differences between the two assays, just to see that you're not losing sensitivity or that you're still getting the responses that you would like to get. There are quite a few publications showing that the trend in terms of responsiveness correlates pretty well between ELISpot and FluoroSpot assays, but there can be sensitivity differences depending on the different cytokine combination that you have. We have a few resources on our website, and if you would like to talk to some of our FAS teams, we can also talk about different kits. But I think it's clear that if you are establishing an assay, you should validate it for that specific combination, and anytime you change anything in your panel, you should revalidate and reassess, right? Do you agree, Aurélie and Sophie, with that interpretation?
Yeah, definitely. And especially when switching from ELISpot to FluoroSpot, I would think it is also exactly the same. In our hands, I must say that we once did a head-to-head comparison of interferon gamma ELISpot versus FluoroSpot, and that we actually had a higher sensitivity in the FluoroSpot simply because we had less background. And especially, I think it can also be due to that very time-sensitive step at the end of the development that you were mentioning, Renata, for the ELISpot, where basically this can be, you can have a lot of variation there, while with the FluoroSpot, the incubation time is fixed, and you just stop your reaction there and add the fluorescence enhancer. So that's, I would say, a plus on top of it.
Yeah, that's also what we've seen from our customers, that It's a bit easier with the FluoroSpot not being a time-sensitive step. And actually, there's a question just on this topic. So for FluoroSpot developing time, will the development time affect the results if the development was shorter or longer? Because in ELISpot, you can see the spots and stop the development. So I can start this question because definitely with ELISpot, it's a visual method because it's you just need bright light to visualize the spots, and you can look at the inverted microscope and visualize the spot development. So that's unlike in FluoroSpot, you need a fluorescence reader to illuminate the wells first before you can see the spots. But yeah, the major difference between the assays is that in ELISpot, the longer you leave the substrate, the more spots you get, but the more background you get, right? And that can contribute to then the slower signal to noise. While in FluoroSpot, the enhancer step is not time sensitive. It's fixed amount of amplification of signal, basically. So you don't have that stress. And so it makes it a little bit maybe easier and less stressful in the lab. So yeah. Okay, so we have another question about what is the upper limit of quantification of each analyte in multiplex FluoroSpot assays? Oh, I think this is a great question. And Aurélie and Sophie, you have done linearity testing, and maybe you can share your experiences of, you have 3 different cytokines, you have interferon gamma, granzyme B, TNF. They all sort of behave a bit different. So what were your reflections when it comes to the upper limit of quantification?
Yeah, and that I think indeed one important comment there is that it's actually can depend on your instrument setting. So you can of course tweak the settings to really reach the upper limit of the machine. But those settings might not be ideal for the number of spots that you're actually interested in, and the more moderate responses that you want to detect. So that's why it's also difficult to say there's one fixed limit or range of upper limit of quantification that we see, because it really depends on the settings that you use. There is probably a real upper limit of the machine that you could test, but how we approach it is actually with Aurélie, of course, to optimize the settings to detect the relevant responses. And then you might come earlier to a limit for those, for instance, CD3, CD28 stimulated controls, where we could really see that almost all the samples had a plateau at the higher cell densities. But that doesn't mean that the machine could not maybe detect higher responses in a linear range if you adjusted the settings, but then you would have suboptimal performance in another range. So that's why with the FluoroSpot, it's not a very absolute upper limit of quantification, but really depends on, or at least partly depends on the settings. Don't know if you want to add.
Yeah, Aurélie, do you have something to add to this?
Yeah, I would say, as Sophie mentioned, usually our primary focus will be on the specific responses that we want to see to measure any vaccine reaction or immunotherapy responses, and this is, I would say, rarely reaching the upper limit of quantification, which is usually more reached for your assay control, on which, as we said during the webinar, we can indeed set acceptance criteria based on the performance of that control, but this should be indeed just an acceptance criteria, just to qualify your run, to ensure that everything went well during the seeding, during the development, and so on, rather than really being your primary focus on such assays.
Yeah, and it's indeed a good point that you bring, maybe, Aurélie, that indeed for relevant responses across very different fields, the upper limit of quantification is never, I would say, a relevant limit, because it's always much higher than the responses that you see in patient samples. Indeed, with these CD3, CD28 stimulation, you might reach it, but that is really only for those controls that are not giving very biological relevant responses.
Yeah, and we have seen that definitely positive controls, if you don't want to seed fewer cells per well, you will get them sort of blown out, and they will get this carpet effect where there's so much secretion that the spots can't be distinguished anymore. And these wells need to be treated specially during the data analysis, and it's best to have a process in place that defines how you handle the settings for those wells and how you report them. But at the same time, if you're using them as a yes or no answer, it's not so important what value you're getting out of them. But you definitely need a process in place to deal with these type of wells, because if they do get if you do get a lot of secretion, and sometimes it does happen that the patient is, you know, a super responder out of nowhere, and then those wells are blown out. So if you really want quantitative data from those wells, it's best to reanalyze the patient with fewer cells per well with the same article, and get a more accurate measure, or report that patient as above upper limit of quantification but positive. So this is how we typically recommend our customers do that. But then lower limit and upper limits are always a bit context dependent, instrument settings dependent, and also panel dependent. So you will have different levels for different kits as well. Great. So I'm just seeing if there's any more questions coming in. One question I have is about the TNF background, and as you showed in your results, and you touched a little bit on that, Sophie, when you were describing the limit of detection, is that different cytokines will also have different levels of background. So I think this is quite interesting, and I'm curious about your perspective on is it a FluoroSpot issue, and have you seen it in other assays, and you know, why do we get this result when it comes to TNF and its biology?
So I don't know already if you want to start, or
Yeah, maybe if you want to start with the statistic outcome that we had here, and then I can continue.
Yeah, so indeed, we see this high precision. We see it only in the TNF alpha, which is a strong indication that it's not purely technical, because if indeed the stimulation of the cells would not be homogeneous across the wells in general, we would see it across the cytokines, but it's really very specific for TNF alpha. It's also known and observed more widely. So we do really believe that it's a biological underlying reason. We indeed, how we can deal with that is if we really want to have good quantitative data for TNF alpha, we have to have sufficient number of replicates per condition. Because a high CV does not mean that your numbers are not reliable per se, they're just imprecise. And with many values, with multiple replicates, the average of those replicates will become more precise. Another way to deal with that is to move away from a true quantitative outcome and more go to a qualitative outcome, because we did see during our qualification, for instance, that we can reliably and consistently still detect responses above background. So it's definitely not that you cannot interpret the results, but of course the high CV is also something that we should take into account when designing then the clinical testing, and also how we interpret the data.
Yeah, and I think the TNF alpha can also be still a very good added value when it comes to looking at polyfunctional T cells, for example, in combination with interferon gamma. Yeah, just to see whether your cells can be characterized as polyfunctional or not. And it's true that it seems high as a background compared to the two other cytokines that we have evaluated here. But if you would be thinking in terms of number of cells, this is just only a couple of hundred cells within the 200,000 cells which were seeded. So that's still very, very low and very sensitive, and even more sensitive in our hands and in our experience compared to other cytokine readouts, such as ICS, for example. So it might seem high compared to the two other cytokines, but still, as Sophie mentioned, it will really still allow you to detect quite nice positive responses with a nice assay range.
Because indeed,
go ahead.
No, I just want to say that indeed, if we compare it to maybe the assay that is used most often as alternative, the ICS, because that is a rare event flow cytometry readout, the variability is also very high, because those rare events are very difficult to quantify very precisely. So definitely the variability there is also very high in general, not even only for TNF alpha, but we see very high CVs there typically.
Yeah, and we know that different cell types produce TNF alpha. We know monocytes can secrete TNF alpha in addition to T cells, and we have seen that that can cause the background levels to be higher. I really like how you highlighted that different cytokines will have different levels of background, and that's just to do with their biology and which cell types are secreting them in response to stimulation. And it's rarely used as a single readout for positivity anyway. So I think added value in combination with our cytokines is really it's really where it shines. So, okay, so we have also another question is, do you validate your assay for different antigens, or your validation is based on model antigens, meaning peptide pools? So basically, whether the qualification data, do you perform it only on the peptide pool controls like Cephros, or do you also do it with the test articles for each study, basically?
Or if I start.
Yeah, I guess you can start. I guess we will go back to the dry runs that we were mentioning at the beginning anyway. So I guess you can start.
Yes. So indeed, I would say definitely the more technical parameters, they do not have to be analyzed specifically per project. Like for instance, the linearity, I can be justified that it doesn't have to be evaluated specifically per project. And for others like sensitivity, for instance, or specificity, we didn't really touch on that. But for that, it can be indeed relevant, as Aurélie already said, to see if there is not a very big background issue and that the test articles give already a response in negative patients. But that depends a bit, but that is more Aurélie's expertise. It's not always possible, for instance, to know for 100% certainty that patients should be negative, for instance, or should be, there's not always already material available for patients that should be positive, that are definitely positive. So it depends a little bit how much knowledge there is already and how much material there is that these additional things make sense. But as Aurélie said, these dry runs, definitely to see that there are no issues with the assay, these are, I would say, standard. And then further evaluations depend a little bit on how much information there is or how much material there is to do meaningful things.
And maybe to add on these dry runs, even if you wouldn't have any patient samples that you could include in these, the idea is also to evaluate whether all the process that is done upfront, the FluoroSpot assay, is actually working well, meaning everything that I was mentioning about just the PBMC isolation procedure, freezing, shipping, thawing, and so on. This can really be evaluated in the dry runs as well, whether there is anything that needs improvement. If you would see that by thawing simply healthy donor sample, but that we're isolating using the same procedure that you will be having for your clinical trial samples, would lead to a poor viability or poor functionality of your cells. This is the very beginning where you need to maybe adapt a bit the procedure that will be done for your precious samples. And this is then something that you can do early on and really know also early on. If you know that there will be a cell-based assay readout included in your trials, I would highly recommend to at least evaluate the procedure of how the samples will be collected.
This is really critical, really, for this assay to work. You rely on high viability and happy cells, basically, because if they're not happy, you will not really get any cytokine secretion from them. So definitely those processes need to be optimized. Great. So I think we answered all questions that came in. Thank you so much for your questions, for those who are listening. And we will reach out after the webinar. And yes, feel free to email any of us if you have further questions. And I really want to thank Aurélie and Sophie for your excellent talks and your insights. And thank you for participating today. So I wish you a good day, and until next time.
Thank you, everyone. Have a good
Bye.
day. Bye.
Thank you.